Oh, I don't know why this isn't working. I'll try it again.
Yeah.
I'm just kidding.
You know, that little story, I remember when I was dating my husband, he was so sick, and he came over with garlic water. I was so disgusted. And then his whole family does that. Did you? I'm glad you enjoyed it. Thank you. On her time. You need that. I'm going to help
Okay, so let's finish with our hearing. So here, so for your test also, there'll be a picture like this. I'm going to ask you to label the external ear, the middle ear, and the inner ear. So just so you know. Also remember, excuse me, I'm going to have a picture of the nervous system and you identify which one is sympathetic and which one is parasympathetic. You know that. That's also going to come out, okay? So same with this one. Using a pencil, you put a bracket. Where's the external ear, where's the middle ear, and where's the inner ear? Okay? In the past tense, I always had pictures. So this is how we hear, and all of this from the external to the middle ear, this is not, they don't contain the nerves. The nerves are always locked up inside, and so they're in the inner ear. You see, this one right here, this is your cranial nerve number 8. And they come out of the inner ear, right? So first, what is the function of the external ear? So the external ear is from your auricle. We know where the auricle ear, this one is, right? Auricle up to the tympanic membrane is your external ear. Is there a function to this one?
It pulls the sound waves in.
Yeah, doesn't it, right? Like your dogs, if they hear something, doesn't the ears go up like that, just like, so that's what our ears do. And so who's that one? Mike Tyson? He has no ear? Yeah. Oh, he's the one who bit the ear, right?
Holyfield.
Yeah, so, you know, if you're, that's very important. And that's why I think you say, what did you say? What did you say? And if you cup your ears, like, because it channels, it harvests the sound waves. Here it's called light waves. Here it's called sound waves to the ear. So it channels them.
Put a glass to a wall, you can hear better.
That's a good idea. I'm going to do that so I can hear what you guys are talking about. Okay, so here in the external canal, what's here? You have a hair over there, right? And you also have sebaceous glands, modified glands that secrete cerumen, or the earwax. Is there a function to cerumen? What's
it
the
collects
function?
the dirt, so it keeps
Yes,
the dirt.
and it's also acidic, right? Any oily, because it's an oil-based secretion, has an acidic pH. It's open. Look at this, is always open to bacteria entering. So the sebaceous glands, the cerumen, the earwax prevents bacteria from becoming pathogenic, overwhelmingly dividing, and cause invasion inside where your nerves are. So if someone says, oh, you got your earwax out there, what you're going to say? Oh, thank you. Right? It's protective. So I'm glad I have it. Right? So then you have your tympanic membrane. Okay? Look at this one. The tympanic membrane, between the tympanic membrane and this one, oval and round window. OR, your operating room. So the OR marks the boundary for the inner ear. Besides in the OR only, it's, what do you call that? No authorized personnel only, right? So the OR is for the inner, only for the inner circle of friends, okay? So external ear up to tympanic membrane, then tympanic membrane to your OR, oval window and round window, that's the boundary of your middle ear. And the inner ear is everything after your OR. So next we go to the middle ear. You're going to write that the middle ear is air filled. It's filled with air. And very important for you to note this tube right here. This is called your auditory tube or your eustachian tube, or another term is pharyngotympanic tube. Okay? So that's why when there's pressure here, you yawn or you open your mouth so that you equalize the pressure, right? And this is also the reason why maybe if you have oral infection, it can also affect your ears. You can have otitis media. So this is the middle ear, okay? It's air-filled. What it contains are the three smallest bones of the body: the malleus, the incus, and the stapes. You know of miso soup?
Yeah.
Okay, so MIS. Okay? So, like, or TMI, we all say that too much information. The tympanic membrane is the one that articulates with the malleus, so TMI, and then after the malleus, incus, and then last is your stapes. And the stapes is the one that touches or is resting on your oval window, miso soup. So the oval window. So it's like the window has openings, right? So the stapes is sort of the opening. It rests on the oval window. Okay? Do you follow? Before we talk about the physiology of hearing, I want to look at this inner ear. The inner ear, where is my ear?
On your head.
The lab exam, I always have this ear out because we don't have any other ear. So there's the ear. So there's your external ear, right? And then, what happened to my tympanic membrane? It's gone. Oh, here's the tympanic membrane, okay? So there's the tympanic membrane over here. So external ear up to tympanic membrane. And then the tympanic membrane is air-filled, and there's the three bones over here. So the TMI, right? Too much. So tympanic, the first bone right here is the malleus, and then the incus, and this is the stapes. This stapes is sitting on my oval window of my inner ear, the oval window of the inner ear, okay? And what did you need to remember about your middle ear? It contains the eustachian tube, so right here. It connects to your oral cavity, okay? So this is your inner ear. I'm going to take it out. So you see the inner ear. Look, the inner ear is inside the bone. You see, it's inside the bone. So there's your bone. There's the bone, right? So it's embedded inside the bone. Okay? So inside the bone, there is a labyrinth shape-like structure. So this is So see, that's why they have this. They cut through the bone, and that's where this one is. And inside this structure is fluid. So the inner ear is fluid filled. Do you follow? Okay? It's just like your tires, right? Your tires have an inner tube. So the inner tube contains the structures for hearing, but it's protected by the bone outside. Like your inner tube is protected by the tires outside. Okay? So here, this one. This houses my receptors for hearing. Okay? It just coils like this. So what I'm going to do, I'm going to uncoil it to see where these fluids are. So first, it's like this, you see? So it's bone, and inside there's this fluid. And so this part right here is the one for hearing. This part right here is for equilibrium and balance. Okay? Over here, this is for equilibrium and balance, detects movement. This is the part right here for hearing. Both of them, they report to this cranial nerve number 8. This is your nerve, your vestibulocochlear nerve. Okay?
Make a great water park ride.
Yes. Excuse me. So let's look at, so what I'm going to do, I'm going to uncoil this so it's easier. So let's uncoil this like, what's that gum that they're rolled?
Bubble
Then
tape?
You uncoil, bubble tape, something, you uncoil it. So I'm going to uncoil this. It's fluid, and it's separated. So there are separate fluid compartments. So you see here, so this was the coiled inner part, right? And I uncoiled it, right? And right in the center, you see this, the organ of Corti. So there's this center part, so there's fluid on both sides, you see? And this center part right here is like a membrane where my nerve endings sit. The nerve endings are in the organ of Corti. So the organ of Corti is for hearing. Organ makes a sound for hearing. That's how I try to remember. Okay? So here on each side is fluid. You see that? There's the window, right? The oval window. There's the stapes. There's my meso, right? E-M-I. Then, meso. And there's a window. So here inside my inner, and look at the fluid, by the way. Look, when I uncoiled it, there's no complete separation, right? The fluid bathes all over. Do you see that? And this is another window called the round window. So the inner ear has two windows. The oval, meso, and then the round window. And it's fluid, two fluids that sandwiches this spiral organ, the organ of Corti. The organ of Corti contains your hair cells, which sits on a membrane called the tectorial membrane. So the hair cells, which sits on a membrane called the tectorial membrane. So there's the, you see tectorial membrane? This is the tectorial membrane over here, right? You see that? So there's the tectorial membrane. You see the yellow? On top of the yellow is the tectorial membrane. Within the yellow is where this basilar membrane is where I find sitting my hair cells. So I have a lot of hair cells right there. The hair cells are touching the tectorial membrane. Okay? So this is what makes up my organ of Corti. It's made up of hair cells. So there's too many. So there's the scala tympani, scala media, scala vestigia. Don't worry about that. it's better for us to understand how we hear. So there's two fluids here, okay? And they say that this is your scala tympani, the scala media, don't worry, scala media, okay? So, but fluid, right in the center, that sandwich between the two fluids is my organ of Corti. The organ of Corti consists of my hair cells, which is touching a roof called tectorial membrane. So it's really touching like this, okay? So this is how you hear. Look what happens when I hear sound waves, right? Do you, when you've gone to the disco, I'm sure you have, you've heard some loud music and you feel, you can feel the vibration, right? Or sometimes you can even see the speaker really being moved by that vibration. It's waves that's doing that. And that's what happens. Those sound waves, it's like a gong. So it's going to hit the tympanic membrane. Can you picture that? Your eardrums. The sound waves will hit that and bang on the tympanic membrane, which will bang against my meso. These are diarthrosis, so they're freely movable, which then will bang against what? The ova, meso, ova window. What will the ova window do? It will cause a wave, right, on the fluid. And so when the wave goes like that, it will then cause movement or touching of the hair against the roof. When it moves the hair against the roof, you generate action potential. Then where is it going to go? Cranial nerve number 8 to temporal. You heard something. You see? Did you get that? So it's that waves that create a movement right here. So that the key is the hair to be bent. To open your mechanical gates, right? You have to bend that hair so that you can hear. You generate an electrical impulse. So the sound waves are converted to an electrical impulse. This is very important for you guys to know. Look at this. I told you that these three bones are freely movable. They are diarthrosis. Okay? So, look at your ears. This is what's referred to as amplification. So, amplifications. The sound waves, look, they are amplified 20 times. Because look at, from all of this, it's being cut and channeled into a small, just like you're converging of the vision, right? So you're converging them all into a smaller area. And if all of those waves are going, the pressure is going to be transmitted from here to here to here, it's possible to destroy your hair cells very quickly. So the normal sound that we hear is a muffled version of the actual intensity of the sound all the time because of contraction of two smallest muscles in the body, your stapedius muscle and your tensor tympani. Stapedius must be contracting, or the one that holds on to your stapes, right? It tenses your stapes. And then the tensor tympani must be the one that tenses your tympanic membrane. You see? And there's your stapedius muscle over here. So these two muscles, when there's sound waves, when you create a gong right here, they tense. So that instead of transmitting all of that pressure to the hair cells, it just muffles it. But because they are so tiny, it's very easy to injure them. And we know that when we injure them, you cannot regenerate that anymore, right? Okay? They're very fragile. How do you injure them? Loud blast? Or like with lawnmower, just lawnmower. They advise you to wear that, what do you call, head cuffs. Here, ear muffs, right? Ear muffs. Or like the, I don't like it when I'm driving next to someone with a boom, loud music in their car. That is dangerous to your hearing. We don't realize that, right? So those kids, when they get to be in their mid-20s, the first sign that your hearing is going is increased sensitivity to hearing. Because this is damaged, and so there's nothing. It just gongs right away, so you're so sensitive to hearing. You think, wow, I am Superman. I can hear better now. No, it's going. It's about, you're going to lose your hearing younger than if it wasn't damaged. Does it make sense? Okay? Even aspirin, all of those NSAIDs, the pain relievers, if you look at the information on those drugs, one of the side effects is ototoxicity. It's toxic to your ears. So if we constantly take all those pain medications, after a while we'll have problems with hearing. It damages this one. Do you follow? So you see, it's very easy. How do we hear? Your sound waves creates a gong right here in the tympanic membrane, right? And then you can trace TMI, right? TMI and then meso. So tympanic membrane, MIS, which steps on my oval window, right? And the oval window will create waves in the fluid inside my inner ear, which contains the organ of Corti, right here. The organ of Corti has the hair cells. The hair cells have a roof called the tectorial membrane. So the waves, like the waterbed, when we lay down on the waterbed, and that's what happens. So how do you equalize that wave? The oval window. The round window. The round window pops out, you equalize the pressure, and you're ready for another round of waves. That's it, very easy, right? So what kind of receptors are your hair cells? Mechanoreceptors, right? Because the waves, the pressure opens it, mechanoreceptors. So how about the last part, the very last part of Bio 241, which is this one? The vestibular apparatus and the semicircular. Yes.
No question. When they use like a cochlear implant for hearing, how does that affect this? How do they actually hear with that?
That's what happens. You need the cochlear implant is nothing but you're taking away the injured part and you're just implanting it as a brand new one. So it doesn't affect it. You still have the fluid. What happens when you can't hear is your hair cells. You still have fluid, but this one, the hair cells are dead. So you want to replace it, right, with fake ones. But the fake one is still functioning as a nerve. So you can see this part right here, they have different areas of intensity. Like there's an area for hearing loud, also high pitch, low pitch. So depending, that's why when you have hearing deficiency, they send you for hearing tests, and the hearing test will tell you exactly what pitch is your problem, and then they can pinpoint it here where the problem is, and so they can go directly in there and put an implant. That's
So they make it work with the magnet and the hearing aid.
Yeah.
Because I know when my nephew takes that off, he can't hear.
What happened to your nephew?
He was about a month old and got sick, and they treated him wrong, and he went deaf. Because it's cute. He's 19 now, and he takes it off when his mother is irritating him. He used to, when he was little, he'd just cover up his eyes because he could sign, and she'd be yelling at him and sign, and he'd just cover his eyes.
So this, by the way, this is the summary of hearing, which is exactly what we just talked about. Okay, slide number 68. So there's your, see, there are the hair cells. They're sitting on that membrane. And so now the last part is, how do we detect moving? How do you know when you rode the elevator, how do you know that that elevator was going up? You can sense it, right?
Yeah.
Or when you're nodding, how do you know you're nodding? Yes, or shaking. All of these movements. All of this movement
It's the fluid.
Yes, it's fluid. But it's not actually fluid this time. Semi-fluid gelatin. That movement, that's the job of this one. Okay? So you have three semicircular canals. So circular, it's a semicircle. So this part right here, we just studied this part, the cochlea for hearing. Now we're studying these circles right here, semicircles, three of them, three semicircular canals inside, right? And then this bulge called the vestibule. So within the vestibule, you have, and the vestibule and the semicircular canals, you have their hair cells again. But this time, the hair cells, instead of fluid, they have a roof made up of calcified gelatinous substance. It's like a jello with calcium. Okay? And so there are my hair cells. So this bulge, first, this vestibule is made up of two parts, the utricle and the saccule. I think it's too much for you guys to remember. Okay? But let's look at first our directional movement. How do I know I'm going forward, backward, nodding, or not? Right? So here in my vestibule, over here, you see, I'm going to go to this. See, hair cells again. It's just hair cells. And the hair cells have that roof, gelatinous roof, called otolithic membrane. And this membrane, because it's gelatin, when you bend your head like this, isn't that gelatin going to slide? When it slides, won't that brush these hair cells? So you generate action potential. Very easy. Go to cranial nerve number 8, do temporal lobe. That's how then I know I'm bending. Or like when I'm moving forward, the gelatin is sliding. So I know I'm moving forward. Right? Okay? And that's what happens when you get car sick, right? When you are car sick, your ear, because this is the one that detects movement and equilibrium, your ear is telling your temporal lobe you're not moving. But then your eyes see that you're moving. And they say that there's a disconnect when you get to the brain, and the brain gets confused, you become car sick. Like, come on, tell me what's going on. So that's why they say when you have that car sickness, you focus on something right way ahead that's not moving. So that your eyes and your ears are agreeing that you're just not moving. So there's no car sickness.
So is dynamic equilibrium, is that in the central canal or the semicircular canals? Or
the semicircular canals, yes, is for dynamic, for movement, right? Rotation, any kind of movement. This semicircular canals, okay? So look at the semicircular, any kind. So dynamic means movement, right? Other one is static. So dynamic is, so whatever kind of movement, right here, I have hair cells again in the bulge of the semicircular canal. So there's a bulge right here called the ampulla, and then there's the crista ampullaris and hair cells again. So whatever kind of movement, there's just always that gelatin. And anytime you move, the gelatin slides and brushes the hair cells to generate action potential. And that's just the physiology of hearing, equilibrium, and movements. And that also ends your nervous system. And Bio 241. So, take out your, it's not bittersweet yet, you still have Bio 242. Oh, when you're done with Bio 242, you say, I am done. Now you just say, I take a break. Then I'll see you again, you say. So, break, use the rest of the time. You know your eyes, you know your muscles, you know your brain, it's over there. Take it out and study because you're, so today and then Wednesday after lecture, you just go ahead and do your muscles again, right? All of that to prepare for your lab exam. And then after your lab exam on Monday, then we do comprehensive review for your final exam on Wednesday. And then you're done. Okay?
Can we do comprehensive on Wednesday?
It is.
So that we have the whole, no, can we do the comprehensive
this
review?
Wednesday.
Yeah, so
No.
that we have the weekend to study for the final? Instead of one day?
Oh, yeah, we can do comprehensive, but you just want to do also your lab. You want to prepare. Some people, yeah, we can do that.
Yeah, that would just give us more time to, for
Yes,
those of us
yes,
that
yes,
listen
yes.
to it.
Yes.
I have a question. With the muscles,
Yes.
when you set out the lab exam, all of these are going to be on the cat, right? You're gonna, if you're going to use them, these are all going to be cat.
Yes.
These are the only ones that will be human, right?
Yes. Yeah. And right there you have your models there too. And they're
Yeah.
not that hard to find.
No, and you know, I found on
Oh.
YouTube has got some great dissection videos where they pull the muscles out, and it's a lot. I can look at a piece of paper till the cows come home.
And by the way, like trapezius has the spinotrapezius, as long as you write trapezius,
The clavia,
that's good.
the acromonial,
Yeah.
and the spino.
But if you just write trapezius, that's good. Trapezius is trapezius. Deltoid is deltoid. Okay?
Okay. Because I was worried when I was studying, and I'm like, in some places they're very similar on the cat, in other places they don't look.
But then, once you really look at them, and you say, oh,
I think
you
the
think
biggest.
it's a lot, but it's not.
No, it's not that much, but I think the biggest difference is if you could stand the cat up and turn his legs in, it'd be closer to a human. But when they're
Yes,
all flattened,
yes,
it's
yeah.
you look at them and you go, because I mean, she's a massage therapist. When we were looking at the muscles, she's like, I know where the muscle's at, but on the cat, it's not the same place.
So then stand it up when you're taking your lab exam, right? Stand up your
We need to be having a partner so somebody can hold your cat up.